glossopharyngeal. Then extraglossal, I'm talking about buccal I'm talking about epiglottis, upper one third of the esophagus, where you taste buds as well. So all these areas, it's cranial nerve 10. So all this diagram, please let it not confuse you. These are just pathways. So you know that in the nodose ganglion, you have the vagus. That's where it passes through. That's its superior, is it inferior ganglion? Then you have the petrosal ganglion for the glossopharyngeal. You also have the geniculate ganglion. But trust you me, in ENT, no one is going to ask you this unless we are dealing with in maxillofacial. Maybe yes, because as they are destroying things in there, I call it destroying because usually they do. They'll be able maybe to affect these, but in ENT, no one is going to ask you this. We are going to ask which of the cranial nerves and then which part of the tongue is innervated by these nerves. Okay. One thing I want to mention, which is important, is two of these cranial nerves, they pass through the jugular foramen, okay, as they are going to the brainstem. So we have the vagus and the facial nerve. These two, they pass through the jugular foramen. And then the coda tympani is just, some researcher says it's part of the facial nerve. Others say it's like a band that holds the facial nerve, the glossopharyngeal, as well as the vagus. They pass through that. Okay. So all that you can add up on your own as you're reading. So into the brainstem we go. Which area specifically? The nucleus of the solitary tract. Where is this place found? In the pons. Part of it is in the medulla. Okay, so just a brainstem, the gustatory area. From the gustatory area, these two pathways, they are at one point they don't cross. They aspire to, but they don't cross at all. Okay, all the two go up, but they don't cross. Remember that. It's different from olfactory. The tracts, they intertwine here and there. So from the nucleus of the solitary tract, we go to the ventral posterior medial nucleus in the thalamus. From here, the signals now can either go, the primary main function is of course taste perception, but it can also go to the hippocampus for what? For store of that memory of the taste. You eat something nice, you still say, I want to go to the restaurant and eat that food, the type of food I ate last time. It was nice. How do you remember that? Some of the signals, as they reach the thalamus, they bounce off to the hippocampus. Others go to the amygdala. Okay. So in this posterior ventral nucleus in the thalamus, if some of them, they reflect and go to what we call the insula, the gustatory cortex, or the anterior insula, okay, in the cortex, frontal cortex of the brain. Are we together? We have what we call adaptation of taste. Adaptation of taste, this means, for example, if every day you eat chili things, like our Indian friends, they eat chili things all the time from childhood. How does tongue or the brain adapt to that? So all sensory systems show adaptation effects. Rarely weakens this stimuli and causes subsequent perception. For example, if you take salt all the time, we take salt, we sweat, some of it is what part of the mucus. So in the mucus, in the mucus component, we test it all the time because we have mucus in the mouth, we have mucus in the nose. These two are interconnected. And then because, how do I put it easier? It does affect us the ability to purely taste salt, like the face time. For example, when the baby face tastes salt, they make a certain face. But for you, you won't make a face. You have adapted to that, okay? Also, another example is what we call cross-adaptation, okay? When the taste of one food affects the taste of another. So if you take a sour beverage, it will taste too sour after taking a sweet substance. For example, those of you who take alcohol, wine, I like wine myself, wine, and then you have chocolate. When you take the sweet wine, of course, it will taste sweet because it's the first thing you've put. But when you take the chocolate and you want to take the sweet wine again, it will not taste sweet. Why? There has been cross adaptation. The chocolate is sweeter than the wine. Okay, so basically, what I want you to understand is that adaptation is that constant exposure to certain types of taste. They will cause either there's certain taste to be reduced or to be more pronounced. Those were just examples too. It's a bit difficult to understand, but it's very easy actually. When you have time, walk through, go through this on your own. So that was that about taste. And then we have certain disorders that affect taste. So absence of taste, where you completely do not taste anything at all, we call it agousia. And then where you partially taste something, we call it dysgeusia. Dysgeusia is, for example, you have had flu for a long time, then you taste food, you just taste weird. Or in pregnancy, when you become pregnant, women who've been pregnant before can attest to this, those that had this symptom. So you find that certain food, you can't taste them the way you used to before. So partial taste loss is called dysageusia. Agousia is complete loss of taste. What can cause this? There are a lot of things. Anything compressing in this pathway. Let's say you have a tumor in the cerebral cortex and the brainstem, Pott's disease, TB in the spine, all sorts of things. As long as any of these three pathways are compressed, you have that condition. I also would like to mention the trigeminal nerve, the third, the trigeminal cranial nerve. This one, literature says it's also involved in taste, but more of perception, things like temperature, things like pressure. I'll give a good example of chili. Chili, it's hot, it exerts a certain pressure on your lips, on your taste buds. So this kind of taste is more of a perception, and it's the trigeminal nerve which is responsible for that. But the three major nerves, we've discussed them. Okay, so we move on to olfaction. Olfaction is basically smell. So if you look at the nasal cavity, you have, of course, the frontal cerebellum, all the structures forming here. So above the nose, in your nose, on the roof of your nose, you have what we call the olfactory epithelium, olfactory mucosa. Okay, this is found in the superior, near the superior nasal concha, in the nasal cavity. So where your nasal bone is, somewhere where your nasal bone kind of terminates, that's where you find below it, you find the olfactory epithelium. So above the olfactory epithelium, you have the cribriform plate of ethmoid bone. Okay, you remember the China says ethmoid bones on the face. So below it, that's where our epithelium is, olfactory epithelium. Above it, you have basal cells. These are where we call them the stem cells of the nose. This is where these olfactory cells regenerate from, okay? Because they are always also regenerating. Just like our taste cells are regenerating, these are our nasal cells or factory cells, they are also regenerating, though at a slower rate compared to the test ones. The test ones takes about up maximum 15 days, but our olfactory receptor cells, they take up to 2 months, between 6 weeks to 2 months, anywhere in between. So the basal stem cell causes regeneration of the receptor cells. Okay. And then above it, you see the olfactory nerve, connective tissue. We have the cribriform. So we just took a part from here. The cribriform of the ethmoid bone. We have the olfactory bulb. So major structure I wanted to mention above the cribriform, we have the olfactory bulb. This is where in the frontal cortex. Okay. So back to our notes. Okay. The olfactory mucosa, it's located on the posterior part of each nostril, on the roof of each nostril. So the nasal epithelium or the olfactory membrane, it's a pseudo-stratified columnar type. It also has those cells that we mentioned. We talked about the basal cells and the Bowman glands, okay, that produces mucus. So it contains olfactory cells which contain cilia. We all know the function of cilia to move mucus along. Okay. It also, in between these are Bowman glands that produce mucus and the basal cells for regeneration of these olfactory cells. So olfactory receptor cells, they are bipolar, like a true neuron. They are bipolar. They have a protrudes into the nasal mucosa. This is where when you smell something, some perfume, it will go straight to the dendrite roots of the olfactory cell. From there, it will go to the cell body, which is olfactory itself. By what mechanisms? By G protein coupled receptor mechanism. Remember, we mentioned if you smell something, we call it an odorant. When you smell something, an odorant comes into your nostrils, in the dendrites of these cells, it will bind there. What will happen is that when it binds there, it will cause an alpha unit of a G protein coupled receptor to dissociate. It will move, it will go and activate the adenyl cyclase. Adenyl cyclase, when it's activated, it will cause phospholipid C to be activated. This in and ATP and these neurotransmitters will synapse. In this way, in this case, they will synapse past the cribriform plate up into the olfactory bulb. We'll discuss that later. So just a review on G protein transduction. So we all know that the olfactory receptors are chemoreceptors, just like taste receptors. And then odorants, for something to be able to diffuse, for you to smell it in the nostril, it needs to have certain properties. Okay, so this odorant, it must be slightly water soluble and lipid soluble. So it has to be water soluble so it can penetrate the mucus to reach the olfactory cells. So it has to be water soluble. For this odorant also to be smelled, it also must be lipid soluble so it can bind to the membrane. Membrane of which cell? The olfactory cell. And then some of these odorant examples we can be camphoraceous. So you've used camphor cream. Those who've used camphor cream, there's a certain unique way it smells. We have ethereal. We know the soil. When it rains, how does it smell? We have the pungent smell. It's very pungent smell. So we're talking about bleach. We have chemicals, cockroach killer. All those things are very pungent. We have musky cell. Most men musky. They smell musky. Don't ask me which one. You know your men how they smell. Okay, are we together? Hello?
Yes, madam.
Yes, madam.
Okay.
Yes, we are.
So you know how you men smell. Okay, you don't know, your women know. I assume we are all adults in here. We also have a
We
floral
do.
smell. Hey, that's good. We also have a floral smell, okay? So flowers, roses, and things like that. Some perfumes are floral. We also have peppermint smell, okay? Eucalyptus, peppermint itself, certain sweets, things like that. Vicks, that's peppermint. So all those are odorants, okay? There are many of them, but these are the major ones you find. All these odorants, for them to be able to be smelled, they have to be water soluble so that they can dissolve through that mucus layer, okay? They have to be lipid soluble so that they can be able to bind before they can dissolve. Okay. So if you look at this, we discussed this partially. I will not talk about the olfactory epithelium. We've already discussed that. We know what's in there. So if you look up from where it says olfactory nerve, okay, these tracts of this olfactory nerve, as they come from these bipolar olfactory cells, they form these tracts, they'll penetrate through the cribriform and go into the olfactory bulb. So what will go there, these are the axons going up into the olfactory bulb. They are the axons that penetrate the ethmoid, the cribriform of the ethmoid bone. So when these axons go up in the olfactory bulb, they will form what we call a glomerulus. Okay, not the glomerulus in the kidney, no, a glomerulus in the olfactory bulb. So if you look where they are above the cribriform, we have where it's written olfactory bulb. There are these green and yellow junction. So this green and yellow junction, if I was able to circle, I would have circled it. This is what we call the glomerulus. I'll show you another slide. So this is what we call the glomerulus. So you can see that this yellow cell above, it has they call it the olfactory bulb neuron. It's what we call the mitral cell, like mitral valve, but mitral cell. So the mitral cell dendrites and our olfactory cell axons, they fuse together, forming what we call a glomeruli. Okay, they're just exchanging synapses so that the odorant signals can go and you can test them. And then now from the mitral cell, which we call the second messenger, it will cause tracts of olfactory nerve to form, okay, that will go to the brain. A better understanding we get from here. Okay. So we just got from our other slide, the previous slide. When you come here, we have what we say the glomeruli or glomerulus has one. So in this glomerulus, you can see this second area, which is a glomerulus. It has pink signals coming in, which are the axons of which cells, the receptor cells, and then it has the dendrites of a mitral cell. Okay, this cell is found in the olfactory bulb together with the glomerulus. They're in the olfactory bulb. We are not showing our cribriform here because we've already known where it is. And then when these two they fuse together, synapses exchanged into the mitral cell. The mitral cell goes on to make a tract. What tract? Olfactory tract. Okay, so our olfactory pathway, we have receptor cells of axons that synapse the mitral cell, forming a glomeruli. They're also forming olfactory tract, which enters at the junction of the midbrain and the cerebellum. So before we go that far, let's go back to our previous slide. Slide 18. Oh, sorry. So where this glomerulus forms, in the olfactory bulb itself, you'll find another cell called granule. Granule as in granules, granule cell. So the function of a granule cell, which is found in the mitral, in the glomerulus here, its function is to inhibit weak signals. Okay, so it inhibits weak signals. So odorants that are faint, they are coming through here and they are weak. This granule cell will produce GABA. We know what GABA, the neurotransmitter, it's an inhibitory neurotransmitter. It will produce GABA, which will work on this mitral cell and inactivate it. So if it's inactivated, how come are you able to smell? So it only inactivates the weak. So it's like it's taking out the unnecessary. Because imagine if you could smell every little thing, you would go mad. We would go mad. So it's like survival of the fittest. Only the strongest of the odorants will go through into the brain to make a response of you smelling. Okay, remember in the olfactory bulb, we have another cell, granule cell, other than the mitral. Its function is to produce inhibitory neurotransmitter. Which one? The GABA neurotransmitter. So when the synapse, they form, when they synapse, they will enter the brain at the junction where the midbrain and the cerebellum meet to form medial and lateral areas. So when you say medial and lateral areas, just look at it as straight from this cribriform, from the olfactory bulb. Sorry, just let me go back to the, where is our olfactory bulb? So straight from our olfactory bulb, you have certain tracts that will go up the medial areas and others that will go lateral. Just look at it as going down from this olfactory bulb. So these areas are what we have here. Okay, so you have this in purple, that's our olfactory bulb. And we know that it's getting signals from from our receptor cells in the nostrils. They come up, they synapse with the mitral cells, the signal is strong, it's not inhibited, it goes through. When it goes through, you will see an arrow which says medial olfactory area. Some tracts will go up here through the olfactory nerve, the first cranial nerve. And then others will go to the lateral olfactory area. Okay. So some of the signals will come here. So the ones that goes medial, these are the ones that are responsible mostly. It's smelling, but they will stimulate your appetite. Okay, there are times when you smell certain foods and your appetite is aroused. Okay, is aroused. Sorry, it's because of the tracts that go to the medial olfactory area, and then to the lateral olfactory area. This is of course the smell, but this is the kind of smell specifically that makes you like the food. Okay, you've smelled it, you haven't eaten, but you like it. Okay, and then we have the other parts of the brain. It can go to the hippocampus, and then the newer pathways, they go to the amygdala. Okay, so remember that from here you can have medially, they go to the medial olfactory area. This area, it's responsible for appetite. You smell something, arouses your appetite. To the lateral olfactory area, you smell something, you like it already before you even eat it. Okay, but also remember, as you can see, all these tracts here, you have some going to the brainstem. So for the ones in the brainstem, mostly, before they even depacitate to go to the brain, they divert to go and stimulate the salivary glands. The salivary glands, the parotid, to produce mucus. So there are times when you smell food, you're salivating. Okay, especially children, even adults do that. You smell food, you start salivating. So those are some of the signals that get diverted from the brainstem. To smell something when you're hungry. So there are those that go to the brainstem. There are those that go to the hippocampus for memory storage. Remember, for test, we said we also have signals other than the frontal cortex. They also go to the hippocampus so that you can remember the taste of, you can remember the taste of your favorite food. So we also have pathways going to the hippocampus from the smell pathway. That's they go together, okay? Because you find that most of these smell pathways, they are all over the place. But if you remember from our test pathways, they are quite specific. They'll go to the ventral posterior medial nucleus in the thalamus. Then from there, they'll go to the gustatory cortex. That's all. But if you come to our smell, it goes all over the place and it intertwines with the taste. That's why you are able to smell and taste at the same time. And that's why when you have chronic sinusitis or patients with chronic sinusitis, patients with rhinitis, which is inflammation of the nostrils, these guys, they have reduced smell, they also have reduced taste. The best example is COVID. When a patient now comes in that I can't smell anything or taste food tastes bland, like I can't taste anything, just send them for COVID first before you do anything. Because two pathways they intertwine, but the taste pathway does not cross. It's the smell pathway that crosses, and it's the one which is all over the place, making it in contact with a taste cell, the taste pathway. Okay. So, other than going to the medial and lateral places, it goes to the lateral areas, specifically the amygdala, the piriform cortex, and so on. The newer pathway, this is where it passes through the thalamus, the dorsomedial nucleus of the thalamus, and then to the orbitofrontal cortex for conscious analysis of odor. Okay, where you can say, what does this smell like? Is it smelling like mint? Is it smelling like floral? Is it smelling like is it pungent smell? Is it something harmful? Okay. And then, we've talked about this diagram. And then we come to some of the disorders of smell. The disorders of smell, we have anosmia. This is loss of smell sensation that is caused by nasal obstruction. Okay. When I say nasal obstruction, we have a condition we call allergic rhinitis. So a patient comes in with allergic rhinitis, they're telling you I'm flowing away, I feel my nostrils are blocked, mucus is just flowing. You look into the nostrils, you will find that there are swellings in there. Most of our students tell me that there is a foreign object, or there's a cancer or a growth. Those are not growths. The most thing you see in allergic rhinitis, you have inflamed and enlarged inferior tabinates. Those are the ones that are close that you can see. Some will have polyps. Polyps can do that. When they have overgrown, they will occlude the nasal passages. This can cause you to lose smell sensation. Okay. Also, nasal mucosa atrophy. Remember, we mentioned that our olfactory cells, they regenerate. Maximum is 2 months. Okay. So as you grow, you find that your skin, wrinkles, everything ages. So do our olfactory cells. They undergo atrophy. So from 45 years going up, your sense of smell is different from that of your child or your young sibling. It's different because of atrophy that you're undergoing. Okay. Others, it can be because of tumors. So where do these tumors are? Mostly in what we call the olfactory groove. So you've seen between the cribriform plate of the ethmoid and the olfactory bulb. So any tumor occurring here, it can cause these signals to be deranged, okay, or to be lost, or not be able to go through nerve compression. So if you have in the axons of the tests, I mean the olfactory receptors, the ones that penetrate the cribriform and go to form synapse in the glomerulus of the olfactory bulb. If these are interfered with in any way by a growth in here, so growths like people who have gliomas or neuroblastomas in children or meningiomas, okay? So these cancers can also cause one to lose smell or cause anosmia. A good example is a glioma. Meningiomas also can occur and cause anosmia. There is also what we call parosmia. Parosmia is partial smell. Parosmia, smelling things that are not there. You find this in psychiatric or early onset of Alzheimer's or any other psychogenic conditions. Anything psychogenic, one of the symptoms a patient can present with, not all the time, but it can also, if it will cause want to smell things around there. Someone will tell you, I can smell the fish, rotten fish, but it's not there. And all of you can't smell it. Only this patient can smell it. Okay, so psychogenic disorders, and it's quite common in psychiatric patients. We also have what we call, hello? Please come again. Hello? You had a question? You have a question for me?
No, madam, you can go on. I was just relating.
Oh, okay. Yeah. So, but I don't mean that you have psychogenic disorders when you have this. It could be anything, okay? And you don't have to label your patients as with psychogenic disorders when they come with this, but that's probable cause, all right? And then you also have what we call cacosmia. This is receiving a very bad smell, not even nice. Oh, and on parasmia, it could be anything. It could be someone saying it's smelling bad, or I can smell perfume, which is not there, or I'm smelling rain when it hits the ground, ethereal when it's not there. It could be any smell that is absent that any other person cannot smell at that particular time. There's also what we call the cacosmia. This one is specifically bad smell. Someone just come and I can smell a pterodine, okay? When it's not there, sometimes I can smell a pterodine, I'm smelling a rat that has gone bad, and things like that. So the most common cause of this is infection of the sinuses. Remember, we have, how many sets of sinuses do we have? Can someone tell me or mention to me the sinuses that we have? Okay, so we have the ethmoid sinuses, we have the sphenoid, we have the frontal sinuses, and the maxillary sinuses. So these are air-filled spaces. They balance your head. Without them, your head would tilt to the back. You would walk around with someone who has hydrocephalus. But because of this presence of these sinuses, they relieve that heaviness, okay? They balance out the weight for the head. So if these places are inflamed, they become filled with fluid. You have what we call sinusitis, okay? So someone who has that chronic sinusitis, they come in with I'm smelling something really bad. The front of my head is heavy. Mucus is always flowing. I have a headache. Just know that you're dealing with what? Sinusitis. You have to be able to help them decongest these areas. You can do it medically by giving a steroid. You give them a decongestant. Mostly we give xylometazoline. Decongest these areas together with the eustachian tube in the ear. Other than that, you give them an antibiotic, because for them to be able to smell, it could be that this fluid which has been accumulating, it has now formed pus, and the smell is coming from there. So you need an antibiotic, a good antibiotic. Most common use in our setup is amoxiclav. They take it for 2 weeks. You give them a steroid. Why do we give a steroid in sinusitis? In this case, we give a systemic one. You want to reduce inflammation, okay? So that these fluids can stop being produced by the inflamed cells. Remember the 5 signs of inflammation? There is edema there. So want to get rid of that edema, you give your prednisolone. Yes.
Sorry, we have a class at 2:30. So I'm just requesting that you'd wind up. Thank you.
Thank you. Yes. Oh, so that was actually, I'm spending a lot of time on it because it's the last one. So chronic sinusitis. Other than that, in lung cancer, okay, someone comes, they have cacosmia, they're smelling something which is not there. It's because of lung cancer. All right. So we've come to the end of our nose and mouth, taste and smell. Okay. Thank you so much for everyone who participated.